So today in this video we are going to talk about various hypersensitivity types reaction. So there are mainly 5 types of hypersensitivity reaction, but in those 5 type of hypersensitivity reaction we are going to talk about the 4 types of hypersensitivity reaction in the detail. So the type 1 hypersensitivity reaction is also known as the anaphylactic and atopic type. It is mainly mediated by the immunoglobulin E. The type 2 hypersensitivity reaction is the antibody dependent cellular toxicity hypersensitivity reaction. The type 3 hypersensitivity reaction is immune complex mediated type of reaction. There is the formation of the antigen, antibody and the complement complexes in the endothelial cells. The type 4 hypersensitivity reaction is the delayed type. Here what happens, there is CD8 plus mediated cytotoxicity, whereas there is also the inflammatory reaction that is caused by the CD4 plus T cells. And the last, it is not available in the books, but it is available in only some literature. So you can search up about the type 5 hypersensitivity reaction. You can search in some articles. Some research has been made for the type 5 hypersensitivity reaction. I will discuss this type 5 hypersensitivity reaction in brief at the end of this video. The type 5 hypersensitivity reaction is the stimulating type. So if you want to know more about the type 5 hypersensitivity reaction, you have to do some literature review and review some of the articles regarding it. Now let's start the various hypersensitivity reactions. Now let us talk about the type 1 hypersensitivity. The type 1 hypersensitivity is also known as the anaphylactic and atopic type. So it mainly caused by the immunoglobulin E. So anaphylactic and atopic type of the hypersensitivity have the two phases. One is immediate phase and another is the late phases. The immediate phase occurs within the minute, whereas the late phase occurs within the hours. So within the immediate phase, within the minutes, what happens that the antigen cross-link the preformed immunoglobulin E on the persistentized mast cell. So there will be the immediate mast cell degranulation and there will be the release of the histamine and tryptase. So this histamine is the vasoactive amine and the tryptase is a marker of the mast cell activation. So they cause the variety of the symptoms, like there could be the bronchospasm, there could be the excessive secretion in our body. So whereas in the late phase there will be the inflammation and the tissue damage. So what happens is that the chemokine in the late phase attracts the inflammatory cell, for example eosinophils and other mediator, for example leukotrienes from the mast cell, that does the inflammation and the tissue damage. Here you can see there is one allergen and you can see the here mast cell. The mast cell has the Fc receptor for the IgE and it has the allergic specific IgE. So in the allergic specific IgE you can see the immunoglobulin E attached. So it is the sensitized mast cell. When the sensitized mast cell encounters the allergen, so from this various reaction there occurs the degranulation. The granules that is present inside the mast cell comes out from the mast cell. Then this process is known as the degranulation. So type 1 reaction is fast. It is anaphylaxis. It can cause anaphylaxis and it is the first type. So the test for this type 1 hypersensitivity is the skin test or blood test. ELISA is present for the allergen specific IgE. Some of the examples of the type 1 hypersensitivity reactions are anaphylaxis, for example food, drug or bee sting allergies, or allergic asthma is also comes under type 1 hypersensitivity reaction. So now let us discuss about the type 2 hypersensitivity reaction. The type 2 hypersensitivity reaction is antibody dependent cellular toxicity. What happens is that the antibody binds to the cell surface antigen that is present in the abnormal cells. So there could be the cell destruction, inflammation and cellular dysfunction. Three things can occur: cellular destruction, inflammation and cellular dysfunction. The cellular destruction, first of all let us talk about this. The cellular destruction occurs when the cell is opsonized, for example coated by the antibodies, leading to either phagocytosis or activation of the complement system. So phagocytosis or complement system attacks those cells and it causes the cellular destruction, and natural killer cells kill those type of abnormal cells by the antibody dependent cellular toxicity. And let us talk about now the inflammation. The inflammation occurs due to binding of the antibodies to the cell surface. This leads to the activation of the complement system and the Fc receptor mediated inflammation can occur. and cell dysfunction. The antibodies bind to the cell surface receptor and the abnormal blockade or activation of the downstream process occurs. So for example, there is abnormal cell, any abnormal cell in our body that present the surface antigen. So this abnormal cell are in abnormal cells present the surface antigen, which is recognized by the natural killer cell. This is natural killer cell. I have already discussed the function of the natural killer cell to you. The natural killer cells has the Fc receptor for the IgG, and when the IgG binds to this Fc receptor, this IgG recognize the surface antigen, it binds to that surface antigen, leading to the cellular destruction, inflammation, and cellular dysfunction of that cell. So what are the various tests for the type 2 hypersensitivity? That is direct and indirect Coombs test. In the direct Coombs test, it detects the antibodies attached directly to the RBC surface, whereas the indirect Coombs test, it detects the presence of the unbound antibodies in the serum. Some of the examples of the type 2 hypersensitivity reaction of the cellular destruction are the autoimmune hemolytic anemia, immune thrombocytopenia, transfusion reaction, hemolytic disease of the newborn. Whereas the inflammation, they are the Good-Pasteur syndrome, rheumatic fever, hyperacute transplant rejection. And the examples of the cellular dysfunctions are myasthenia gravis, Graves' disease, and pemphigus vulgaris. Now let us discuss the type 3 hypersensitivity. Talking about the type 3 hypersensitivity reaction, the type 3 hypersensitivity reaction is the immune complex type. So what happened? The immune complex, mostly the immune complex of the antibody in the hypersensitivity type 3 reaction is the IgG. So the immune complex, antigen antibody complexes, there is formation of the antigen antibody complexes which ultimately activate the complement system. These type of antigen antibody complexes, you can see it is the antigen and these are the antibody. So this type of antigen antibody complexes get deposited into the surface of the endothelial cells. So this after the deposition of the surface of the endothelial cell, this antigen antibody complexes activate the complement system. So after the activation of the complement system, this attracts the neutrophil. And after the attraction of the neutrophil is recruited in those antigen antibody complexes, and those neutrophil will release the lysosomal enzymes. You can see the lysosomal enzyme, the enzymes from the neutrophil, mostly the lysosomal enzyme, it damages the intercellular cells by the inflammation and various other processes which ultimately cause the tissue damage of those endothelial cells. Now let us discuss about the various examples of the type 3 hypersensitivity reaction. The examples are the SLE, polyarteritis nodosa, and post-streptococcal glomerular nephritis. So these are the main examples of the type 3 hypersensitivity reaction. Now let us discuss about the serum sickness and the Arthas reaction that are the part of the type 3 hypersensitivity reaction. So serum sickness is the prototypic immune complex diseases. Here what happens, same mechanism occurs. The antibody to the foreign proteins are produced 1 to 2 weeks later. And antibody antigen complexes form and deposited in the tissue. This causes the complement activation and inflammation and the tissue damage. The various clinical features of the serum sickness are fever, urticaria, arthalgia, proteinuria, lymphadenopathy, which occurs after the 1 to 2 weeks after the antigen exposure. And there could be the drug associated with the serum sickness also, and that drug is mainly the penicillin. And various infection can also cause the serum sickness. The infection is the hepatitis B. Now let us discuss about the Arthas reaction. The Arthas reaction is the local subacute immune complex mediated hypersensitivity reaction. So what happens when there is if we do the intradermal injection of the antigen into the pre-sensitized, which has already the circulating immunoglobulin G individual, it leads to the immune complex formation in the skin. This is also called as the enhanced local reaction. So usually we do it to the booster vaccination, and it is the cause, it is mainly the clinical features of the Arthas reaction are edema, necrosis, and activation of the complement. So the type 3 reaction you can also remember is at the 3 component. It has mainly 3 component, that is antigen, antibody, and complement complexes. So this is type 3 hypersensitivity reaction. Now let's discuss the type 4 hypersensitivity reaction. Going towards the type 4 hypersensitivity reaction. So the type 4 hypersensitivity reaction is the delayed type of hypersensitivity reaction. So here what happened, there are two mechanism of the hypersensitivity reaction, each involving the T cell. First is direct cell toxicity. It is direct cell cytotoxicity. Here what happened, there is the involvement of the CD8 plus cytotoxic T cell. Here CD8 plus cytotoxic T cell kills the target cell directly, whereas there is inflammatory reaction, there is the involvement of the CD4 plus T cells. Here what happens, the effector CD4 plus T cells recognize the antigen and release the inflammation inducing cytokines, which is shown in the illustration. So here what happened, there is the antigen presenting cell. In the antigen presenting cell, there is the MHC molecule which presents the antigen to the sensitized T helper 1 cell, and from the sensitized T helper 1 cell there is the release of the cytokines. So these cytokines activates the macrophages, and these macrophages, these activated macrophages are mainly the root cause of the inflammation. So we already know that the type 4 hypersensitivity reaction is the delayed type of hypersensitivity, and it doesn't involve any antibody. So now I am going to give the various examples of the type 4 hypersensitivity. One is contact dermatitis and another is the graft versus host disease. So various tests that is done for the type 4 hypersensitivity reaction, or the examples of the type 4 hypersensitivity reaction are the protein purified derivatives PPD for the TB infection that we do is also the type 4 hypersensitivity reaction. Also the PAS test for the contact dermatitis, and the candida skin test for the T cell immune function. So these are the examples of some of the tests of the type 4 hypersensitivity reaction. So so this concludes our type 1, type 2, type 3 and type 4 hypersensitivity reaction. Again, I am going to revise that the type 1 hypersensitivity reaction is the anaphylactic and atopic, whereas type 2 is antibody mediated. The type 3 is antibody, antigen and complement system complex, that means immune complex mediated. And the type 4 is the delayed cell mediated hypersensitivity reaction. So there is one type of hypersensitivity that is known as the type 5 hypersensitivity reaction. So this is not found in books or any step 1 first aid book, but some literature review also gives the type 5 hypersensitivity reaction. You can look into some literature regarding this type 5 hypersensitivity reaction. Here what happens is that the antibodies are produced with the property of the stimulating specific cell targets, or stimulating type of hypersensitivity reaction is type 5 hypersensitivity reaction. It is given in some of the literature only. You can search in some literature. So the example is here it is given the Graves disease. So the Graves disease is the only example I found for the type 5 hypersensitivity reaction. However, you may look up into it. But the Graves disease also comes under the type 2 hypersensitivity reaction. But whenever in the exams or whenever there is the MCQs, is that in which option the Graves disease falls, you should choose the type 2 hypersensitivity reaction unless it is given the type 5 hypersensitivity reaction. So in the Graves disease, what the Graves disease is caused by the antibody that stimulates the TSH receptor, leading to the overactivity of the thyroid gland. So it is the stimulating type. So it in the new literature it is categorized as the type 5 hypersensitivity reaction. So I hope you understood this hypersensitivity reaction and their types. Thank you.